Hi, it's Dr. Barr, and I love me some histology. We're going to talk about the blood-brain barrier. By the end of the session, I want you to be able to correlate the histologic structures that comprise the blood-brain barrier with their locations, functions, and mechanisms of macromolecule transport. Let's discuss the blood-brain barrier, which develops early in the embryo. There's interactions between glial astrocytes and capillary endothelial cells. It's a functional barrier that protects the CNS from fluctuating levels of electrolytes, hormones, infectious agents, exogenous substances, and metabolites circulating in the blood vessels. It's much tighter control than most tissues. The barrier is created by tight junctions fixed between endothelial cells. Keep in mind that most capillaries in the body are fenestrated type, whereas in the blood-brain barrier, it's a continuous type of capillary. For those of you who need the visual, here we have our capillaries bound to each other via tight junctions right here. Then you have your endothelial cell basal lamina, which is this pink membrane right here. Occasionally around capillaries, you will also see pericytes. The key feature here is also the astrocyte processes, which actually are attached to the basal lamina, aka basement membrane. Things that can easily pass through the blood-brain barrier include oxygen, CO2, and lipid-soluble molecules. Neurons are very sensitive to high potassium extracellular fluid concentrations, so astrocytes at the blood-brain barrier will buffer the potassium to maintain its concentration. Too much potassium actually can kill cells, so that's why it really needs to be maintained extracellularly. Those that cannot pass through the blood-brain barrier are substances that have a high weight. In order to get these large substances through, they would need to have active transport. There are also endothelial cell plasma membrane proteins that can help metabolize certain molecules, such as drugs or form proteins, that will prevent the crossing of these molecules. Let's provide some clinical correlation. Dopamine cannot cross the blood-brain barrier. So in order to treat patients with Parkinson's disease that require, because they are dopamine deficient, we would need to give them levodopa or L-dopa, which can easily cross the blood-brain barrier instead. Herpes encephalitis is a virus that can gain entry through the blood-brain barrier and subsequently would affect neurons. Multiple sclerosis is a dysregulation of the blood-brain barrier, which causes, again, migration of leukocytes and release of cytokines into that region of the CNS, causing myelin and axonal damage. Thus, here's a recap of the blood-brain barrier. It protects the CNS from various substances. It maintains a boundary with continuous type capillaries and those tight junctions. And then the astrocyte foot processes are attached to the endothelial basal lamina, supporting communication between the CNS environment and the blood flow. On that note, contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye.